Okay, so I'm going to give the introductory talk to this school, and it's going to really serve as an introduction. There's quite a lot of material which we gave to you beforehand, just to glance through the slides. The aim really is to give you some snapshots of research in the area and to bring you all very roughly up to the same kind of levels, because you all have such different backgrounds, okay? So I'm not expecting you to immediately understand all of this material, but you will come back to it during the summer school from the various talks, okay? Right, so an overarching perspective on chemical neuromodulation. So, rather controversially, I'm going to say that we're in the age of the connectome. What is the connectome? Well, it's a description, essentially, of all the connections in the brain, of its structural organisation. And this is being pursued from a variety of perspectives in humans, but also in other animals, using a variety of techniques, including, for example, graph theory, and also the new neuroscience tools which are enabling us to collect this information about the connectome. And this first slide shows you a few depictions, if you like, very schematically, of connectome type stuff. There's some brain imaging analysis here. This is real anatomy, this is the real brain. Okay, through the cortex, you can see these neurons. And this is, well, this is connectionism. This is neurocomputational brain, okay, which is also based on a connectome-like structure. But what I'm going to argue, and I think others will probably argue in this school, and indeed the entire rationale of the school, is that this is going to be an insufficient account of the brain, even if you can link the connectome to functionality. In other words, to understand what different networks in the connectome fulfill as behaviours or cognitions. I think that's going to be insufficient, and that's why you're all here, and that's why you're also fascinated by dopamine. So, as we know, the cortex, this is the human brain, this is the human reward system. Of course, it's a network, including the nucleus accumbens, or the ventral stratum, and the head ganglion of the orbitofrontal cortex. But these networks and these sectors of the brain are under what we call chemical neuromodulation. And these are some of our famous neuromodulators, some of our favourite ones. Dopamine, noradrenaline, as I'm going to call it, not norepinephrine, this is Europe. Serotonin, or 5-HT, 5-hydroxytryptamine, and acetylcholine. Now, these are just some of the neuromodulators, but they are perhaps the best studied. So inevitably, the school will focus on these systems. Okay, so what is the interaction between chemical neuromodulation and the connectome? This is it. What are the functions and implications of chemical neuromodulation? So the argument that I'm going to put forward is that these systems are providing a variety of states under which the connectome functions. And these states include, well, states like states of arousal, wakefulness, alertness, states of motivation, if you like, mood, reward, to use a much maligned term. Stress. Is stress one thing? Are there different types of stress? And all of these systems, therefore, are modulating the connectome to work at different levels, at different states, at different degrees of optimality. That's the argument. So immediately you can see how much more complicated this will become. We're not just studying a static connectome, we're studying it in a variety of states. And for the computational person here, I'm using states in a different way than computational scientists think of it, as stimuli. This is a difference between psychology and computation. but these are states. And the argument is that these states probably evolve very basally from conditions of metabolic need of organisms and the need to avoid harms. And most of these systems have evolved in that context. And so the other argument is that what neuromodulation can confer also is the ability to reconfigure the connectome in a variety of different networks. So this is a speculation, but there is some evidence to support this. So again, one enhances the overall adaptability of this structure. Okay? So that's the touchy-feely kind of rationale of modulation. So what is meant by modulation? So I've got two definitions here for you from two scientists I admire very much, Floyd Bloom, who's written an outstanding book on the biochemical basis of neuropharmacology. And in one of his essays, and he's written several essays, Floyd says, neuromodulation is that set of neurochemical processes that curtail or prolong, augment or diminish effects of fast signaling in neuronal networks, in other words, in the connectome, often conditional on the firing state of the postsynaptic cell. So we're talking about something which is very sophisticated. It's modulatory. Okay, it can enhance or diminish, it can curtail or prolong, and it can go either way, depending on the state of the system. Quite sophisticated. And another sophisticated view is by Dreyer and others, and this is for the computational approaches, which you're going to hear about, particularly from Michael Hasselmoes here, parameters and equations for artificial neural networks that slowly change the dynamics of the network. It's an interesting definition. So these are two of the definitions I've found, and maybe you can find others. So what is chemical neuromodulation? What it isn't is fast signaling through ion channels, glutamate, GABA. It's not that. It's not direct. It's not the on-off switches of the connectome or the network. It's the modulation. It's the control exerted on this transmission via biasing inhibitory excitatory influences. And neuromodulators are affecting presynaptic release or postsynaptic response to another compound acting as a transmitter. So they're adjusting the effects of the transmitters. It's typically slow and spatially diffuse. You may have heard of volume transmission. So these substances don't generally work on a very fast timescale, rather intermediate to slow, and they diffuse over wide extrasynaptic spaces. They don't just work tightly at one synapse. So this is another important principle. Mediated by GPCRs, G-protein coupled receptors. This is very important. Almost, I think, entirely neuromodulation is mediated in that way. And these GPCRs actually have a variety of functions. They change the biochemical properties of the recipient nerve cell so that it changes its hyperpolarization, for example, or its propensity for action potential. But they also have longer lasting effects to alter genetic transcription, for example, in the neuron, and produce much slower time changes, which may take many weeks or days to occur. So there's this complexity of the direct modulation and then the very slow overall modulation through genetic means. Okay, so just to give you an example, a very quick snapshot of this biasing effect, and to celebrate Floyd Bloom's contribution to the area, this nice experiment carried out in the dentate gyrus of the hippocampus in a rat anticipating food, a hungry rat, with a CS, a tone signal, auditory tone. Initially, the tone is not paired with food, it's irrelevant. And if one looks at activity in the hippocampus, if anything, it's inhibited. Then in their experiment with Siegel, he electrically stimulated the locus coeruleus, presumably releasing noradrenaline, although we know nowadays it may be doing more than that, and I'll come back to that. And as you can see, it inhibited further the activity to the tone. Okay? So it's a modulation, enhancing the inhibition. Then the tone was made salient by being predictive of food, and suddenly the cell becomes more interested in this. Yeah? No longer inhibited, mildly excitatory. Now we stimulate the locus coeruleus again. Wow, it's augmented. So the same effect has different actions dependent on the activity of the postsynaptic system. When it's interested in the tone, it's enhanced. When it's not interested, it's made less interested. So in some sense, it's enhancing the selectivity of responding to the tone. Maybe a function of selective attention. So that's the kind of thing we mean by neuromodulation, graphically. Now, I mentioned GPCRs, and there's about 360 genes which code these, I think, in mammals, as opposed to about 70 that code ion channels. So that's the other main means of transmission. And here's a taxonomy of neurotransmitters. Why are there so many? And how did we all get there? These are questions I've been asking myself. This is just a list, and I'm not going to go through it in detail, but GABA and glutamate, well, it's interesting, you know, these are both side products of Krebs cycle, the respiratory cycle, so probably that's how they evolved, okay? They're metabolic products. They've mainly worked through ligand-gated ion channels. And now we have the first problem, because actually sometimes they can be neuromodulatory. So glutamate can be neuromodulatory under some circumstances. So this hard and fast definition, as usual, in biology and neuroscience doesn't quite work, okay? And when you come on to the classic neuromodulators, the monoamines, and I'm kind of including acetylcholine in this, it's an honorary monoamine, first to be discovered, of course. Generally, again, GPCRs, but of course acetylcholine also works through nicotine receptors, which are fast signaling. So there isn't a hard and fast definition in terms of transmitters. It depends on the receptor system. Adenosine's a neuromodulator, and probably adenosine was just about one of the first biochemicals when life originated. So presumably that's why biology used it as a messenger to produce adenosine, which we understand all too little about as a neurotransmitter. There's the endocannabinoids and then the peptides, which really are multiple amino acids. These guys are all derived from single amino acids, but enkephalin, for example, has 5 amino acids linked up. So this comes from a giant genetic super producer, as it were. And by studying these superfamilies of GPCRs, new neuromodulators are constantly being discovered, and one fairly recently is orexin or hypocretin, which I'll say a few words about. They often act as co-transmitters, so we have the example of cholecystokinin and dopamine in the same neurons. They're released together. We're not quite sure what that does. CCK seems to affect how dopamine works. Neuropeptide Y and noradrenaline, vasoactive polypeptide in the cortex and acetylcholine. I'm sure Martin will tell us all about that. So we don't really understand what the function of co-transmission is. Sometimes these substances act as hormones and sometimes as transmitters. Ghrelin, an eating hormone, is an example. And of course then we blend into the general area of hormones which are released into the general circulation and which aren't going to be the main subject of neuromodulation. Okay? So there's a lot to consider. And we have a lot of tools to study them, and I think we have to use all the tools available in order to gain understanding. And here are the main tools that I can think of. Obviously anatomy, neuropharmacology, because we have a variety of drugs which can affect these substances. Single unit electrophysiology, population electrophysiology, very important. I'll say a word about optogenetics and chemogenetics. Psychophysiology, mainly in humans, GSR responses, galvanic skin response, heart rate, EEG measures, and then a variety of psychological approaches. And after all, the function of the brain is cognition and behaviour, so we have to measure that particularly carefully and understand behaviour. And neuroimaging, a whole range of modalities, and James Rose here, and I'll introduce him, Cambridge, and Roshan, and also Imre Duzel and Gita Muznudsen are expert neuroimagers who will tell you about the whole range of modalities for neuroimaging, which we're using in humans and in animals to some extent to understand the macro functioning of these systems. So optogenetics, don't let me explain this to you. You have Zachary Mainen, who's going to be describing it, but this amazing discovery of Carl Dizeroth with his opsins, which you can put into particular cells with molecular tagging, genetic techniques and viruses, absorbed into the membranes, and then shine a light on the cell or its projection and activate or inhibit via these different opsins the particular pathways. So this is an amazing level of sophisticated selective responding. You can see the difference between what I just told you about electrical stimulation, where you're non-selectively stimulating a range of systems which may be very different, whereas with this specific system you can achieve selectivity. Also with chemogenetics or DREADS, which I know some of you are using, and hopefully we'll have a discussion about DREADS, which is analogous to optogenetics, but it works over a longer time course. And instead of shining light on these receptors, you have a ligand which is inert other than for these receptors. And when you give it by mouth even, in theory, it stimulates again just that pathway. Wonderful. And clinically, as James may tell us, interesting for the future. But a lot of technical issues. So behaviour, as I said, is also important. And one thing I wanted to tell those of you who don't work very much with the behaviour, we can measure behaviour very carefully, measures of memory, so-called executive function by which we control behaviour. In humans, a variety of methods, including computerized methods, and in animals. So we have analogous means of measuring working memory, cognitive flexibility. I heard someone interested in this interesting concept, cognitive flexibility and attention in various ways in animals and humans. So we'll meet some of these methods during the course of the summer school. And those of you who aren't very expert in this area, I'm sure myself or Martin, Sato or others will be able to give you some background to these methods, some advice. So evolution of transmitters, I'm not going to say very much about this. You have the slides, but a lot of these things are present in very simple animals. I think you go down to sponges where a lot of the sponges have the genetic apparatus for neurotransmission, but they don't have proper nervous systems. So they have all the elements, but they haven't put them together yet. And it's interesting to track some of the evolutionary aspects. Noradrenaline tends to come a bit later, interestingly, and this is true for development in the mammalian brain. The noradrenaline neurons develop after the serotonin and dopamine neurons, so ontogeny recapitulates phylogeny in that case. And here's a little slide, obviously outlining the main phyla, just the dopamine system, and you can see how, well, there's phenylalanine hydroxylase and tyrosine hydroxylase. This is where these enzymes are first seen, and then some of the receptors, because obviously the evolution of receptors has been crucial, and these modulators have a variety of receptors which have sometimes completely different functions. And so it's a very, very complex system we have to bear in mind. Remember, in the vertebrates, they're the first ones who have basal ganglia, so the direct and indirect pathways of the stratum first occur there, and this is relevant for the distinction between D1 and D2 receptors, for example. So some investigators focus exclusively on invertebrates, and this is just to scare you all. This is a slide from Eve Marder's work. She mainly works on the crab stromatogastrid ganglion, which is a very complicated thing. And she studies neuromodulation. And these are some of the modulatory neurons that she's discovered. And they all work together to produce these different modes of action of the stromatogastrid neurons. The fast pyloric rhythm, the slower gastric mill, or the slower cardiac sac rhythm. They have different rhythms in this animal. And they're all produced by different configurations of the neurons caused by changes in their neuromodulatory system. So if you want to study a simple, complex system, this is a good one to start with. Personally, I've always felt that I have to make my work relevant to humans, and so I like to work with mammals, which are a bit closer to humans. I even work with humans. But obviously, this is an important approach. And this is just to highlight the fact, all the elements one needs of to have a neurotransmitter or neuromodulator pathway. At the top here, actually, I've got the constituents of the octopamine pathway, because octopamine is used as a neuromodulator instead of noradrenaline in insects. So in the invertebrates, octopamine is much more prevalent than noradrenaline. So noradrenaline is a relatively modern invention, although it is present in some invertebrates. And you can see what we're talking about here is the fact that I'm sure most of you are aware of, that most of these amines are synthesized from dietary amino acids, tyrosine and tryptophan. One needs various enzymes to produce a biochemical cascade to result in the transmitter. Then there's a whole variety of mechanisms for regulating the optimal level of the transmitter, including transporters, an noradrenaline transporter, the dopamine transporter, which essentially regulate the amount of monoamine in the synapse so that there's not too much. They, in other words, bring the amine back into the presynaptic neuron. And then a whole set of catabolic mechanisms, monoamine oxidase, catechol-methyltransferase for oxidating or methylating these transmitters and sending them away into the bloodstream. Okay, so these are very complicated biochemical systems. And they're all concentrated in these classic neurons, these classic synapses. This is my own favourite transmitter at the moment. Actually, I've liked them all from time to time, but actually at the moment for me, serotonin is the one. It is so complicated. And here's the presynaptic and postsynaptic neurons. Tryptophan, as you see, we can study serotonin function in humans by depriving humans of tryptophan transiently, and this reduces the amount of serotonin in the brain and produces quite interesting effects. I don't know whether Roshan or others might discuss this, but there are ways of using these systems to study their function. And obviously 5-HT itself, when released, activates a variety of receptors. I think there's about 16 5-HT receptors. 5-HT7, we know someone's interested in 5-HT7 receptors here. They're also autoreceptors, and this is very important, not only on the presynaptic terminals, so that they actually regulate release. They're inhibitory, so they're very sensitive to the transmitter and will inhibit its further release. But these autoreceptors also occur way back here on the cell bodies, so that the system can regulate its own activity, essentially, so that it releases transmitter at the level of the cell body, and this regulates whether it makes an action potential or not. So these systems are under very tight control. And they were originally discovered using histochemical fluorescence methods, which of course have now been evolved much more. There's many ways now of studying the anatomy of these systems, but this is the classic work of Falk-Hillarp and a range of other Scandinavian investigators. Dopamine cells in the nigra, noradrenaline cells in the locus coeruleus, and 5-HT cells in the dorsal raphe. See different shades of green and yellow, so quite tricky sometimes to discern to the non-expert. And these have led to maps in the rat brain. These are sections of rat brain of the main systems, schematically shown here. And I'll just point out some interesting features. So noradrenaline, there's really several cell groups, but two main cell groups from our point of view. The dorsal noradrenergic ascending bundle, which essentially innervates the entire telencephalon, forebrain. There's cortex and the hippocampus. This is very much implicated in cognition. And the ventral ascending bundle, which mainly innervates the hypothalamus. Both of these structures innervate the hypothalamus, but the ventral system more so. And parts of the limbic system, and the ventral bundle not as well studied as the dorsal bundle, probably involved in vegetative states, including feeding, for example. There's also two systems at least with the cholinergic system, and Martin's going to give you a 3-hour extravaganza on the cholinergic system today, so I won't labour this, but we have the basal forebrain neurons, including those going to the hippocampus. I tend to clump these together. But we also have this fascinating tegmental group, which importantly innervate the thalamus and other structures, and involved in awakening and arousal. And similarly, serotonin, we have two systems, a dorsal raphe system and a ventral system. The serotonin system rather intriguingly complements the noradrenergic system, grossly speaking. And in general, the serotonin system seems to antagonize all the other systems. That's one way of thinking about it. Opponents is an important concept in this area, but it's far too simple because it also cooperates with these systems as well under some circumstances. And they're your favourite, the dopamine system. I'm not going to say anything about it, except mainly innervates the stratum, but also in the rat, the frontal cortex, and not the rest of the cortex, although in humans and primates this extends back further. So there's not much dopamine in the visual cortex in humans, but there's quite a lot of dopamine in the temporal lobe, for example. So don't let's think it's just frontal. It's a cortical system. And there's some fascinating things you can glean from this. For example, noradrenaline barely innervates the basal ganglia. So there's only one main projection for noradrenaline systems to the basal ganglia, and it's to part of the nucleus accumbens. So there are fascinating facts like this which show a major principle, which is that there's specificity within all this non-specificity. That's almost a mantra. There's a lot of specific stuff here, but it's quite complicated and hard to understand why this is the case. Why did it evolve like that? These are the human systems. They're remarkably conserved. Of course, it's very speculative because we know much less about the human anatomy, but what we can see generally agrees with primate work and also the rodents. This is the dopamine system again, your favourite. I'm just showing you how it's evolved across the vertebrates and the excellent conservation humans. You can study this at your leisure, but birds have dopamine systems and a ventral tegmental area and so forth, very interesting. And here we have the human brain. And just to point out, of course, that these systems are implicated with a variety of what I call neuropsychiatric disorders. So the frontal lobe system probably implicated in ADHD, also in schizophrenia. Patients with schizophrenia probably have altered frontal dopamine systems, as well as subcortical systems. Parkinson's disease, patients have problems with cognition, which James Rowe is going to talk about, and perhaps Roshan. Then we have in the reward system, which I know you're all dying to hear about, drug abuse, antipsychotic drug effects possibly, is where they work, because Parkinson's movement disorders are in the dorsal stratum largely, and you'll hear a lot about that. In fact, a lot of you are interested in this area, as I understand your abstracts. So again, James will give you a masterclass on Parkinson's disease, I'm sure. So there's a lot of translation to be done of this work from animals to humans, and a lot of inspiring targets to aim for here. This is true of the other modulators as well, of course. So I have it in my contract that I have to mention histamine. So your organiser, Menno Witter, said, make sure you mention histamine. So here it is. Histamine is a transmitter as well. It's a very important transmitter, and it has functions in sleep and memory formation and arousal, and here are its receptors. So don't forget histamine. It's also extremely important, as is Orexin. I mentioned Orexin earlier. So Orexin is an amazing neuromodulator, really. I mean, it's recently discovered, but obviously extremely important, mainly based in hypothalamic areas, but it sends neurons all over the brain, and it does seem to regulate each of the other systems which I've mentioned, acetylcholine, dopamine, noradrenaline, serotonin, generally in a positive way. I mean, this neuromodulator is an arousal agent, and it's very much caught up in the regulation of sleep and waking. And we all know the phenomenon of narcolepsy, which is probably an underactivity of this system, where, and as you know, in narcolepsy, do you know what narcolepsy is? Yeah. So narcolepsy is when, if you fall asleep in my lecture, no surprise. If I fall asleep during my lecture, that's narcolepsy. Okay, so that's orexin, but also extremely important and a new target for drug companies to consider. They project all over the brain again. Yes?
So multiple lines of orexin mice have been developed, but they don't really have a phenotype. Do you think it's because the other
Compensatory, yeah, and actually it's a really important concept which I'm going to mention briefly later, but compensation with this orchestra of substances is obviously highly feasible, isn't it? You can imagine, and all these systems are interacting all the time, and again, this is a point I'm sure Martin will make, as soon as you start talking about manipulating one system, there are bound to be effects in other systems. And so this is the challenge that we have to address in the future.
About this, I was wondering about your opinion in terms of, we are considering neuromodulation as neurotransmitters and hormones, actually, and working together, those.
Yes.
But still, I'm wondering if we, in a way, we should consider the chemical properties. I mean, still, hormones and neurotransmitters, they have chemical differences, so the counteraction of those should be on the base of a chemical.
It's a thin dividing line we have between, as I said earlier, between neuromodulation and neurohormones, essentially. But we are drawing this distinction because otherwise we could have a summer school which lasted 4 months if we had to consider all the hormones as well, which have very specific functions often. So I think it's a good point, but let's focus on what we have. So how do these systems plug in to the cortex? So this is a pyramidal cell, and this shows the dopamine D1, D5, and some of the 5-HT receptors in pyramidal cells in the cortex. And I'm going to say a bit more about those later on, but you'll notice that they're present on pyramidal cells, but they're also present on interneurons, and they can affect transmission in a variety of ways. So the D1 receptor in the cortex seems to prolong currents produced by glutamate, by NMDA receptors, and at the same time it enhances transmission at inhibitory GABA-A receptors, with the result that it both boosts or prolongs a signal, but also resists distraction from competing columns. So it's going to have a boosting effect on the signal in the cortex. So we have to learn more about the syntax by which these modulators are actually affecting neural activity to enhance information processing. And that's the kind of approach that we need to look at. But this slide also makes the point, this is work quite old now from David Lewis at Pittsburgh, using a variety of staining methods to show that there's heterogeneity in the way these things plug into the cortex. I'm going to show you a number of examples of this. So tyrosine hydroxylase has great activity in area 9 of the prefrontal cortex. Now why is that? There's a gradient here. So dopamine is much more important in this area than, say, in other areas in BA25, for example. Whereas noradrenaline is also important in these areas and less so in other frontal areas. These are different Rodman areas. These are sections through the rhesus monkey brain. Acetylcholine is fairly standard, fairly level throughout the cortex in this particular parameter. Serotonin, as you'll see, is much denser in more ventral areas of cortex, and you can see that the serotonin transporter molecule is stained here in red, and you can see it's in great abundance in the ventral prefrontal cortex, the orbitofrontal cortex, and the insula. So why is that? Why are there these differences? Again, the specificity within this non-specificity. These neuromodulators aren't all innovating the same areas to the same extent. What is the grammar? What is the logic of this? It's true of noradrenaline and serotonin. So this is classical work from the 80s of Morrison and Foote, showing that although noradrenaline and 5-HT, you can see evidence of fibers and receptors all over the cortical columns, 1 to 6 here, they do congregate in different areas. So in the visual cortex, there's a preponderance in the pyramidal cell layer, the output of the cortex, Whereas for 5-HT, there's a predominance in layer 4, which is where the thalamic input, the sensory input to the cortex, actually plugs in. And of course, if you're interested in LSD and those kind of hallucinogenic drugs, this is just the kind of focus that you may need to have to understand where it works in brain to produce changes in visual perception. So take the locus coeruleus. So locus coeruleus, I don't know, a few hundred neurons in the rat, many more in humans, thousands possibly, but originally locus coeruleus was thought to be the classic arousal system in that all of these neurons, which are noradrenergic, collateralize and they send their branches all over the brain. So you can have a cell originating locus coeruleus which goes to the cortex and the hippocampus. Okay? So it's like an alarm bell for the same areas, the locus coeruleus, an arousal system. But it turns out that, yes, there is some of that, but there's also specificity in the locus coeruleus. And, you know, different regions of the locus even seem to project to different areas of brain. And there's a paper in my reading list which shows how different parts of locus coeruleus project to the motor cortex and to prefrontal cortex with quite different effects and functions. So again, specificity within non-specificity. How do we study locus coeruleus function? The classic ways with electrophysiology. You have one of the great pioneers of locus coeruleus electrophysiology speaking later this week, Gary Aston-Jones. And originally he was among those who showed that locus neurons are most active during the waking state. They're very quiet during sleeping, so again, classic arousal stimuli. They respond to novel stimuli, they like novelty, but they're dumb. They don't really distinguish between different orientations, so they don't have spatiotemporal specificity. They respond to stress, so again, classic arousal stuff. And he actually studied the, where are we? Excuse me, there we go. He studied their activity in a behavioural situation in the rhesus monkey in a task measuring vigilance or sustained attention. I'm not going to give you great details, but he showed an interesting relationship here. He showed that when the monkey was doing best in this task and detecting all the targets, the locus neurons were firing phasically, fast in bursts, phasic firing. But when the monkey began to be distracted or rather troubled in the situation, it was getting rather irritated with sitting in this chair all this time, this activity disappeared and was replaced by rather tonic activity, which was related to poor performance, which you also saw if the monkey wasn't attending at all, where the system was very inactive. So this generated, against performance and activity, this inverted U-shaped function, which is a common motif in this area. An optimal level of arousal for performance, the so-called Yerkes-Dodson Principle. Here it is in all its glory. The idea that level of arousal or stress, if we can call it that, relates to efficiency in some sense of performance, the speed or accuracy of performance. And there is an optimal level of this to produce this state. And so noradrenaline and other neuromodulators will modulate this U-shaped curve, which may also change as a function of how difficult the task is. Now, Roshan Coles is going to give you a modern interpretation of this and show you that this old idea is probably in need of modification, but it is a recurrent theme in this area. Too much activity is bad for you, too little is bad for you. There is an optimum. How do you get that optimum?
I was just wondering, since we were talking about how these neurons project mostly, everywhere in the brain, so is this kind of activity correlates with the amount?
That's very smart, and I'm going to show you exactly what I think you're asking me. That these functions may also differ in the brain in different areas. That's what you wanted to ask me, and so that's what I'm going to tell you about in a moment. Now, how do we get those functions? Well, in terms of the improvements, they're almost certainly these adjustments of signal-to-noise ratio. So this is early work of Jonathan Cohen, who's a computational neuroscientist, who suggests, for example, that one of the things the catecholamines may do is sharpen this psychophysical curve, okay, to produce a stronger signal, inhibiting the low levels, so dampening down noise, making the thing more salient at low levels when you reduce the noise, and then boosting the signal here so that it is expressed above the normal noise level of spontaneous activity. So this is enhancing the signal-to-noise ratio via mechanisms similar to what I described earlier. And here's some work, this isn't my work, but it's summarised in this article which you have, where they use the technique in a working memory paradigm for monkeys of squirting noradrenaline or dopamine onto neurons while they're recording from them. This technique is called microentophoresis, and you can measure the activity produced by the neurons resultant from this modulation, rather direct modulation of the neurons, under a behavioural circumstance. And Amy Arnsten has done work which suggests that, although I don't think this is completely convincing, that noradrenaline boosting at the 2A receptor increases signal processing, as you see here, for the preferred direction where the animal has to remember where the stimulus was as against to the noise, whereas dopamine tends to decrease the noise here in this particular example and thus allow the signal to shine through more clearly. So obviously you can achieve an improved signal-to-noise ratio either way, by reducing noise spontaneously or boosting the signal. And these transmitters may work in complementary ways, but it's not totally clear. I think there's evidence in the cholinergic system that it boosts the signal. Certainly nicotine has that property maybe, but maybe Martin will tell you more about that. Now, there are different receptors for noradrenaline in the cortex, and Arnsten showed that some agonists will improve this working memory task, where the monkey has to remember where the peanut is. But other agents, other receptors which are active at higher levels of noradrenaline, actually impair performance. So the alpha-2 receptor, when stimulated, seems to boost performance, certainly in aging monkeys, whereas the alpha-1 receptor, when you give this drug, it makes performance worse. And she's interpreted this again in the context of the inverted U hypothesis. Too much noradrenaline is bad for you.
It's just a question to working memory in general, actually, because we're looking into cognitive function in humans.
Yeah.
So working memory is hard to define. So typically, as you see here, it's like remembering a specific location of somewhere, something. But the definition of working memory is the ability to store something, manipulate it.
And act on it, manipulate it, yes.
Yeah.
And so there is a way in which we have to translate this into the animal area. So the argument that Goldman-Rakesh would make, the late Patricia Goldman-Rakesh, who really introduced this method in this particular area, is that it is working memory because the animal detects a stimulus and uses it to guide its action. That's the idea. But I think that's a somewhat controversial point. It's rather different. So a test for working memory in humans would be, remember this phone number. This is my phone number, 365878. Okay, you all got it? 365878, yeah? Can you all repeat it to me? Very good. Reasonable levels of intelligence. That's not working memory. That's the short-term buffer. Working memory is, repeat to me my telephone number backwards. 365878. Go. That's working memory. So that's the point you're making. Thank you. We will return to it. So the interesting thing, coming to your point, is that noradrenaline has different effects in different parts of the brain. So what's good for the frontal cortex is bad for the amygdala. And what's good for the amygdala is bad for the frontal cortex. So under high levels of stress, activating these alpha-1 receptors, working memory is impaired in monkeys. Okay? But the same drug at the same doses will improve aversive conditioning in rats by working in the amygdala. So I think this is a very important point because it illustrates what I was saying earlier about the importance of different states. In the state of stress, you want the amygdala working very well to remember how to escape and to avoid. working memory, thinking about plans for the future is less important. In the lower level of arousal state, that is more important for the future. So this is the idea which Amy Arnstein has developed, which I think is interesting. Now here's some other evidence, actually from our lab, showing that dopamine and noradrenaline and serotonin have different functions in the frontal cortex in tests of cognitive flexibility. I'm not going to go into this in great detail, but there are several forms of cognitive flexibility. One is measured simply by reversal learning, where you discriminate between two visual stimuli like these. One is positive and is rewarded, and the other is negative. Simple decision task. Actually, the ultimate decision-making task, I think. This is good, this is bad, okay? Now reverse. What used to be good is now bad, and vice versa. So you have a conflict. It's a decision you have to make. Do you go on the history, or do you go on what just happened? Interesting dichotomy. And similarly, you can make it more complicated. You can have them attending not to objects, but to dimensions within those objects. Is it colour, or is it shape you have to attend to? So you look at this thing, it's colour that's important, but now it's shape. And so that's another form of cognitive flexibility. And these two forms are mediated by different sectors of the frontal cortex. We've shown this in monkeys, marmoset monkeys. The orbitofrontal cortex is interested in reversal learning, which is rather primitive decision making, and the ventrolateral frontal cortex is interested in conceptual attentional set shifting, this higher order shifting. And what's very interesting from our perspective is that, remember, serotonin is very dense in these ventral regions. Well, when we manipulated serotonin in these areas, we reduced it with a neurotoxin, we found these animals were very perseverative. They were inflexible. So when they were punished for repeatedly choosing what was previously rewarded but is now punished, they continued inappropriately to do so. So serotonin has some role in cognitive flexibility in that area. But intriguingly, it has no effect whatsoever on this higher order attentional switching. Whereas manipulating the catecholamines in this ventrolateral area, noradrenaline and dopamine, affects the higher order conceptual flexibility, but in our hands, not reversal learning per se. So there's some selectivity at the level of the frontal cortex in what these systems are doing. Yeah.
Does the learning rate of reverse cues changes if it's, or gets better if it's from visual to auditory cues, or vice versa?
Yeah, from visual to auditory would be an extra dimensional shift, so that's a different type of shift. And that's measured also in rat work, where they get rats to shift from touch to smell. So that's a higher level shift. What we're talking about here is switching from one smell to another smell. Okay. Do you see what I mean? Now, just to follow this up a little bit, somebody called David Moralak has shown rather similar effects in the rat using this task. So thank you for introducing it. I anticipated you. So rats here have to show cognitive flexibility, but they have to burrow through this sand in order to get food, rewards. And the sand is either coarse or fine, but it also is perfumed differently. So the rat has to make a decision. Do I attend to the somatosensory feeling of the sand, the touch, or the smell? And so you can do the same experiments in rats as you can in monkeys and humans by looking at switching between modalities or within modalities. Do you see that? So you can study reversal learning and shifting again. And what he did was to use a drug. So this is the pharmacological approach, if you like, to studying neuromodulation. Quite sophisticated. He used a drug, an autoreceptor antagonist, atipamezole, which works at the level of the locus coeruleus to actually inhibit the inhibitory receptors there and boost activity in the locus. So the noradrenaline system's working faster, and he was able to show that this drug improves learning. So the animals learn to switch much more quickly, as you see in fewer trials following the drug, in a second, okay? However, if he then blocked beta receptors in the frontal cortex, where we think this shifting is computed, then this blocked the effect completely. So the argument is that it's mediated through noradrenergic receptors in the frontal cortex. Yes?
Are these autoreceptors always working following the mechanism that you previously described? So it is located presynaptically to sense release?
These receptors are on the locus coeruleus, and they're inhibitory, the alpha-2 receptors. And this drug antagonises those, so produce a boost. Now, you have to check that with electrophysiology and neurochemical measures, but, in general, this is shown. And the hypothesis that this is mediated by increased noradrenaline activity is tested with this beta receptor antagonist in the frontal cortex, and it blocks the effects. So this is a remarkable study. And this group did another study which again backs up the distinction I made using this method of dialysis. Now, I know some of you from Oxford are using in vivo voltammetry to measure neurotransmitter release, and we've got Mark Walton coming later to describe that method. Another method for doing this, which is slower but neurochemically selective, is that of dialysis. So in dialysis, one uses a probe implanted into the brain, one samples the extracellular fluid, and then takes that fluid and puts it onto an HPLC column to measure the specificity of amine release in that effluent. Okay, so this is a way of measuring, sampling the turnover or the release of the transmitters, perhaps in a behaving situation or in response to a drug.
So blocking the noradrenergic receptors affects only reversal learning, or even during the first training period, it would affect the learning of the task?
We'll have to come back to that later, okay? So it affects extra-dimensional shift learning in the Moralac study, okay, when given before the session where they test the shift. We'll come back to that later. Okay, so using this method for sampling transmitters, they actually measured serotonin, right? Now, serotonin, remember, was implicated in reversal learning, and as you can see here, animals who were exposed to chronic cold stress are very bad indeed at reversal learning. They're bad at some other features of the task, but this is the really key deficit, as you see here. They take many more trials to reverse. So this is consistent with this idea about serotonin and reversal. And in fact, if you measure 5-HT with dialysis in the frontal cortex, you can see here how in the cold stress animals, serotonin is depleted during performance of this task, notably including the point where reversal is happening. So clearly, serotonin is implicated in reversal learning, and if you give these animals a drug which boosts serotonin, citalopram, it's an antidepressant, it improves their reversal learning. So serotonin is implicated in this behavior. Yeah?
What happens if you depress serotonin and increase noradrenaline? Because we saw that that is also involved in this same task.
It's an interesting experiment, but I don't think that's been explicitly tested to do both at once. Let's do one at a time. But you're making the important point that when you change a transmitter, what happens to the rest of the system? And that's an interesting issue, a very interesting issue. Serotonin again, so this is a point I wanted to make as a general principle that these dumb systems like the locus coeruleus and the raphe are regulated not only by input from the periphery, but also top-down from structures like the prefrontal cortex. They send projections to the locus coeruleus or to the vicinity of the raphe nucleus, and this means that processing in the frontal cortex, which is a high level of cognitive function, can actually help to regulate your subcortical systems. That's an interesting idea. And this is shown in a brilliant experiment by Matt and others in Nature Neuroscience about 10 years ago, where they showed that in rats who are made helpless by taking out of action part of their frontal cortex with a drug called mucimol, which enhances GABA and inactivates this structure, it made these animals perform as though they were helpless rats. In other words, rats who are exposed to unpredictable and inescapable stresses or shocks. Those animals often show elevated monoamines and eventually then depleted monoamines, which correspond to states which have been compared with depression or high levels of stress. And so taking out this ability of the animal to realise that it has mastery of the situation, so in other words, these rats who are able to avoid the shocks by pushing this wheel, if the frontal cortex is inactivated, they lose their ability to realise that. And this results in a dysregulation of their serotonin systems. So this very cognitive information about how you have mastery over your world if you do actions and produce effects, outcomes, that is actually affecting the activity of your neuromodulatory systems. It's an astonishing concept. And it's all due to the fact that these systems are regulated in a top-down way.
Is that learned helplessness?
Yes, it's exactly, it's learned helplessness, exactly correct. So there's other ways of studying these systems. I'm just quickly going to run through this. So I mentioned you can use neurotoxins to try and achieve some depletion. You can remove a lot of the noradrenaline system by injecting a drug called 6-hydroxydopamine, which is taken up like the wooden horse of Troy into the neuron and destroys just dopamine and noradrenaline neurons. It's quite a crude technique, but it produces, if correctly used, massive depletions of these transmitters in cortical areas, as you see here, as compared to when it's injected into the ventral bundle. And we and others use these kind of preparations early on to try and study some of the functions of these systems. Now here's a way of tackling the cholinergic system. Quite a difficult system it proved to be in the early days to manipulate with toxins, but using an antibody, saporin, immunotoxin, we're able to remove cholinergic neurons from the level of the basal forebrain here, as you can see in this stained section. And using these kind of procedures, we're able, in this attentional task, to show really different effects of these treatments, suggesting different functions of these systems. So Martin's going to talk much more about this general phenomenon, but the lost acetylcholine can impair attention rather specifically. So can the loss of noradrenaline neurons under certain conditions of distractibility in this case. Mesolimbic dopamine didn't affect accuracy very much in this task of attention, but did affect the speed which animals responded at, their motivational level. And intriguingly, serotonin depletion affected none of these measures, but it did boost the animal's tendency to be impulsive and to respond before they should. So you have these dissociations produced by manipulating these systems in different ways. But this semi-clean kind of rather specific story is cast into doubt by new work using the new tools. And so a particular puzzle has been the fact that people have found recently that manipulating dopamine in the hippocampus can improve memory at D1 receptors in rodents. This is amazing because the hippocampus doesn't get much dopamine. The ventral tegmental projection is very sparse. So how is it that dopamine in the hippocampus can improve memory? In fact, most of the projection for dopamine goes to the ventral hippocampus, which is not implicated in things like spatial memory, which is what the hippocampus does, among other things. So this has recently been addressed in a number of studies. This is a very recent one from the Candell lab, where they showed rather brilliantly, using the neuroscience tools, that there is a projection from the locus coeruleus to the dorsal hippocampus, which probably releases dopamine as well as noradrenaline from the same neurons. Whereas the projection from the ventral tegmental area only goes to the ventral hippocampus. So there are two different projections which may use dopamine in the hippocampus. And surprisingly, one of them is from the locus coeruleus. And when this projection is stimulated using optogenetics, in the spatial mazes they employed, again, improved performance resulting from activation of those systems which were blocked by drugs blocking dopamine receptors. So clearly, there's more complexity here. The same system is releasing noradrenaline and dopamine, and in this case affecting memory, which is a very, very important discovery in human terms, for example, because there was no obvious basis for this previously.
Did they measure dopamine release in the hippocampus?
Yep, they measured a number of, read the paper which is on the reading list. Very impressive. Yes.
Do you know which kind of deformation receptors?
You're interested in locus coeruleus, aren't you? So you would be interested in this, yeah. Yeah.
Which kind of neurotransmitters?
D1 receptors, D1, D5 receptors in the hippocampus.
No, the effects of noradrenaline in the...
Yes, there are effects. Well, they released noradrenaline, but the noradrenaline release was not responsible for the improved performance. Okay. Okay, so the dopamine system. I think you all know about the system you know and love. Point I made earlier is that it innervates both the direct and the indirect pathways, and these are under differential modulation by D1 and D2 receptors, which is, I think, extremely interesting. Whether this system works in an opposed fashion or in a cooperative fashion or possibly both. This is a very intriguing question currently. And we all know the dopamine system is the reward system. So we come to the very important point here that here's an example where animals will choose to regulate the activity of some of their modulatory systems. That's an amazing idea when you think about it. They can themselves detect the optimal level for these systems to function at. And the evidence for this originally is based on work by Bartley Herbal, who showed that rats will inject amphetamine directly into their nucleus accumbens. So this is the classic self-administration paradigm. Normally the drug's injected into the vein, but in this case, directly into the brain, tiny quantities, aliquots, containing amphetamine to release dopamine. And we were able to replicate and extend this study in our own work, showing that if we co-administered dopamine blockers, the rats would respond to this by increasing the level of amphetamine self-administration. So, in other words, they were regulating the optimal level of dopamine in this area. So this is dopamine as a reward transmitter. Now, I don't think any of the other modulators are directly self-administered, unless you count methencephalin or opioids or something like that.
That's after when the animals learn that they might get them.
The animals have to learn this. Yeah, they learn it. Yeah, they sample these two levers. One of them produces drug, the other produces placebo, and they go for the drug. Okay. Classic paradigm. And not only is this rewarding, this dopamine, but it also affects learning. And maybe Michael Hazamo will say a bit about this, the notion of reinforcement learning, and this discovery of Wolfram Schultz in the 90s, that these neurons will respond not only to reward, but they respond to conditioned stimuli which predict reward. So with training, the activity moves back from where the reward's delivered to the presentation of the conditioned stimulus. So the monkey is anticipating the reward. And moreover, if the reward is omitted here, then the neurons stop activity. So in some sense, the system is modelling the occurrence, this anticipation of the goal, of the reward. And this set of effects is already captured in the so-called Rescorla-Wagner learning equation, which we've got here, which is basically saying that there's a mismatch between expectancy and outcome. And learning occurs when you make mistakes, when what you anticipate is wrong. That prediction error helps you to learn. And so this is another major function of this system. So you'll hear a bit more about that, I'm sure, during the course of the school. It's been done in humans with brain imaging. This is a famous study. They used the bold response, again with similar tasks that the monkeys had, although with money, and showed that modulating dopamine, either blocking it or stimulating it with L-DOPA in humans, these human volunteers, seemed to modulate this function quite a lot. And in the brain, they showed that these prediction errors were correlated with changes in activity, and guess where? The ventral stratum here in particular. So this is a beautiful translation of the monkey to the human work. And it shows this possibility of going into a neurocomputational area. Your own work on this with a D2 antagonist, a very selective one, showed some interesting effects. We similarly showed the same result, except that we were able to show this dose dependently, so that we measured how much drug was in the plasma of individuals, and those subjects with the most in the plasma showed the biggest deficits with the D2 antagonist. And interestingly, it didn't affect learning as much as the asymptote at which the subjects reached. And this is also true as another example of what you can do in this paradigm with a genetic polymorphism. We had subjects with the so-called TAC1A polymorphism who have reduced D2 receptors in the striatum. These are normal subjects. Some of you may have this. Okay? And those individuals were more sensitive to sulpiride. They showed the bigger effects when they had the polymorphism than when it was absent. So this is an example of how genetics, even in humans, can affect the neuromodulation. And this is our modelling attempts on this using conventional modelling procedures, and you can see for us the D2 antagonist didn't affect learning rate as much as the choice of the between the two stimuli when the subjects had initially learned the basic discrimination. So we think that the D1 receptor is much more involved in learning than the D2 receptor, and we need to test that hypothesis using a D1 receptor antagonist. But how can we do that? Because the drug companies have these and they don't let us use them. So here's a block politically to science, because we can't obtain a D1 antagonist to test this hypothesis. So this idea that D1 receptors are involved in learning, they potentiate NMDA receptors, as I said earlier, and they work in the so-called upstate of these systems. They enhance LTP, whereas the D2 receptor actually works on a different state of the dopamine system and probably involved in performance rather than learning directly. So we introduced this concept of phasic and tonic firing in dopamine neurons and noradrenaline neurons earlier, and Tony Grace will tell you more about this. So the dopamine system also, like probably the cholinergic system and we think the serotonergic system, all have these different modes of responding phasically or tonically. The exact functions of these modes we don't really know, but of interest. Obviously, phasic is much more involved in direct information transmission. But we can measure tonic transmission with dialysis, and this is an example, again, from our early work showing that dopamine in the accumbens is greatly increased by amphetamine in rats who've been isolated from weaning. So these animals have much more dopamine as a consequence of their social adversity. And that can be measured with dialysis. And it's the tonic background levels of dopamine that we're measuring here, okay? So this may have a function, we're not sure. Well, tonic dopamine can affect cognitive functions, as we saw in this classic experiment of Goldman-Rakesh, where again she used neurotoxin, 6-hydroxydopamine, into the frontal cortex and had huge impairments on the working memory task, which weren't shown in young monkeys for noradrenaline or serotonin, so this is very selective. And in this early study, she was able to show some improvements in these monkeys by giving them dopamine agonists. So this study is very historic, 1979. This is the dawn of the cognitive functions of dopamine. Dopamine is not just involved in movement or reward, it may have cognitive functions. It's a very important idea. And this is work with Ritalin, which releases dopamine and noradrenaline and improves working memory type tasks in normal humans. These are Cambridge undergraduate volunteers, in fact. And we measured improvements in working memory, the reduced errors with Ritalin, as a function of activity of this frontal cortex to parietal system. It actually reduced the activity of this system and made it more efficient, presumably by increasing signal-to-noise ratios. And interestingly, individually, these people, these volunteers, showed the biggest boost in cognitive function with Ritalin if they had the lowest baseline level of working memory. So this is another concept I wanted to introduce, which Kane Roshan will examine further, that very often with these systems you can boost low levels of function or behaviour, but impair high levels of function. And again, it follows from an inverted U-shaped function. So there's a baseline dependency in the effects of these drugs. And you can see it with impulsive responding in this task. If the rats respond too fast, the high impulsive rats show reductions with Ritalin, whereas the low impulsive rats actually show increases in impulsivity. So this is a bit like ADHD, attention deficit disorder. Ritalin improves high impulsive, but it may impair normal impulsive. Again, why this occurs, we're not sure, but it may relate to the balance between phasic and tonic activity in the system. So L-DOPA, as you see, improves working memory in Parkinson patients in this task. So when they're tested on L-DOPA, they do much better than off L-DOPA. This is quite a selective effect, so it was predicted by the monkey work and shown in Parkinson's disease. But L-DOPA can impair some functions. So reversal learning here, actually Parkinson patients did better off of L-DOPA. So the same patients could be improved in working memory, but impaired in reversal learning with the same dose of L-DOPA.
Wasn't that the same with the previous slide? Didn't this say off? The upper curve was off, right?
Yeah, so they did better on. So they made more errors. It's errors, yeah. So they did worse off. Here they're doing better off. And we think this is somewhat to do with how dopamine is lost in Parkinson's disease. So it's probably lost more in circuits which control motor and some, for example, working memory functions, and less initially anyway in those systems controlling reward, so that when you give L-DOPA, you can titrate these systems up to a normal level, but maybe overdose these systems. And this may lead to anomalies such as gambling in Parkinson patients, which has been observed. So these are things, again, which come from this inverted U-shaped type idea applied to different regions, and Roshan will tell you about some updates in this thinking. So genes like COMT, which manipulates the amines, also can affect frontal dopamine. I'm not going to go in great detail about how this hypothesis works, but in the frontal cortex, dopamine is mainly cleared by catechol-methyltransferase rather than monoamine oxidase. And so in the frontal cortex in particular, if you have alleles which make your catechol-methyltransferase more efficient, that will lead to a depletion of dopamine in that area. This is in those subjects, normal volunteers with Val-Val alleles, whereas if you have the MET alleles, you have a less efficient enzyme and you increase your dopamine levels. So in the normal population, it's been hypothesized by Weinberger that you will see changes in cognitive function as a function of their genetics, of their catechol-methyltransferase enzyme. It's quite an amazing prediction when you think about it. And there is some evidence to support this, and again it devolves around this type of function, that those subjects with MET alleles tend to show improved working memory performance and also improved aspects of cognitive flexibility, whereas those with low dopamine in frontal cortex tend to show impaired working memory. So there's another genetic modulation of this system which we need to take into account. And this has been capitalized on in rodents with drugs which block catechol-methyltransferase, and as you see here, improve the ability to shift between smell and touch in this rodent switching paradigm. So again, this is another way of studying the system using genetics. Okay, so we're coming close to the end, and I thought I would just finally talk a bit about recent developments in human imaging using sophisticated means to understand functional connectivity in the brain. And this is going back to quite an old study, but it's being done in more modern ways these days. This is a study by Jenny Cool, where she had subjects in a PET scanner and measured the activity of the cortex in terms of its metabolism, while they were performing a sustained attention task, a bit like the rat task. But in this task, subjects had to attend to this central location and repeat the digits that were presented at a very fast rate here. So you couldn't be distracted at all because you would then make many attentional errors. So you had to sustain attention over several minutes and respond to specific sequences of digits with a response, okay, these particular series. And if one gives clonidine in this task, which is a noradrenergic agent, it affects attention. And what they did was to study the effects of clonidine in the resting state, where there was no task, where the subjects were just lying essentially with their eyes open, versus when they were performing the task. And the point I wanted to make is that you can then apply quite advanced computational methods to look at the correlations of activity in all the different brain regions to see whether these are proxy measures of the functional connections between these systems. And what she discovered basically is that clonidine produced completely different effects if the subjects were resting versus whether they were performing a task. In the resting state, these were the main effects discovered. Changes in effective connectivity, the locus coeruleus to visual cortex and parietal cortex and thalamus. All of these areas were inhibited, so the normal relationship was reduced by clonidine. But when they were performing a task, a completely different state occurred. And here you can see locus coeruleus boosting the parietal cortical activity and also affecting these other frontal cortical regions. And in fact, further analysis suggested that this boosting effect was actually a function of the frontal cortex affecting the locus coeruleus to produce this effect. So these relationships are discovered by modelling, efferent connectivity, advanced kind of mathematical manipulation of these matrices obtained from human brain imaging. So this is the effect, the prefrontal cortical modulation of this particular pathway. So this is the kind of level of resolution one can aspire to in humans, which we can then relate in a more cellular and neuronal way to other animals, with using obviously more interventional techniques. So this is actually her PET scan showing the locus coeruleus and thalamic changes following clonidine in the task. And very recently, this kind of approach has been used using a different mathematical approach called graph theory, and using a drug called atomoxetine, which is again a noradrenergic agent, which again clearly produces different effects in the resting state versus when the subjects are actually doing the task. So producing changes in the way this system is configured, going all the way back to what I said in the opening parts of the talk. So James Rowe, I know, is going to talk a bit more about this methodology in humans for understanding these interactions between different systems as a function of neuromodulation. So just finally, we're just 5 minutes late, and I think we started late because you're all a bit late, so I'm more or less on time. I don't think Martin will mind if we delay coffee by 2 minutes. So I'm just going to go through 4 summary slides, the basic principles of neuromodulation. I'm sorry for this race through, but the other talks will be much more relaxed, but it does give you some basis on which you can work, as it were. So here are some principles modified after Peter Dayan's very stimulating paper in 2012 in Neuron. So the neuromodulatory systems can rapidly report selective information as well as global signals. The selectivity in the non-selectivity, as it were. These signals are produced over multiple timescales, phasic and tonic, and they're often relatively slow but also spatially diffuse because of volume transmission. Different receptors can respond selectively with different affinities and time courses. This makes things very difficult. When you block a D2 receptor, do you have more activity at the D1 receptor, for example? We have to consider these complexities, even within the system, let alone in interactions with other systems. The different receptors are localized in different anatomical regions, which all have different rules for how they compute different information. Autoreceptors are important both on the terminals and on the cell bodies. So nothing's easy. If you give an SSRI, an antidepressant drug, initially it turns off the serotonin system, and then with chronic use, turns it on. So how do we grapple with that? That's difficult experimentally. Then interactions, opponency, but it's rarely symmetric and there's often cooperation, so nothing is simple, although everything interacts. Feedback loops, I mentioned, from top down to these nuclei provide top-down regulation, which of course will be included in high-order information processing regulation of those systems. We've got spiking, phasic spiking and tonic activity. There's co-release with other transmitters, glutamate, but also peptides. These neuromodulators are often involved in energy utilization as well as information transmission. So, for example, neuromodulators are vasoactive and affect blood supply in the cortex, which is a problem of interpretation when you do pharmacological fMRI imaging, as James Rowe will probably explain. They can affect learning and developmental plasticity. 5-HT is particularly involved in development as well as modulating behavior. Neurogenesis affected by serotonin, apoptosis, axon branching, dendrinogenesis, all affected by neuromodulation. But at the other end of the spectrum, they affect key cognitive computations such as working memory. They affect oscillations. We haven't said very much about that, but these transmitters are affecting the way neurons synchronize their activity and produce oscillations. We need to understand that more. The classic example, maybe Michael mentioned it, acetylcholine in the theta rhythm. They affect configurations of how networks function, both in the lobster and the crab, but also in the human cortex. So we've seen examples of that. We have an inverted U-shaped function, but maybe this needs to be modified. We have baseline dependency. We have huge individual differences as a result of experience, social adversity, for example, but also genetics. Produces a range of possibilities for psychiatric disorders. They are major therapeutic targets. Most of the drugs we have in psychiatry affect most of these systems. They need to work better, of course. And there's some further reading from papers which are a bit more obscure, which I was able to check during the preparation of the lecture. And my collaborators are here, James and Roshan. Roshan no longer works with me directly, but James is still in Cambridge. He has his own group, of course, and also our animal work led by Barry Everett, Angela Roberts and Jeff Daly. Thank you very much.